Thank you. Thank you for the introduction. Thank you for giving me the opportunity to speak. And I want to go back a little bit because I think I presented this slide in 2019 at the Microbiome Cozy in Basel. And I don't know how many people in the room today were there. But this is what we were doing back then. We were looking at the global microbiome, looking at all of the data that was available. And at the time, 10,000 metagenomes was a big number. And what we did was we took this by taking data from the ENA, and we assembled it and predicted all of the open reading frames, and then clustered them in these different levels, starting with this species level unigenes and then broader protein families. And so this was what I was presenting in 2019, but probably something I didn't mention it, although, and it was in the paper, but it's a very small sentence in the methods, is that the small open reading frames, we just ignore them, or we filter them out either right at the beginning or throughout the analysis. We just, but back then already we were starting to think that, okay, maybe we shouldn't be throwing out all of this. And so I started to, let's go back to these data or to this approach and look at these small open reading frames. So in the meanwhile, we have more data. So if you see, we have a lot more dots on that plot, and in particular, we're starting to have dots in parts of the world that were really undersampled before. So before we'd have a lot of dots in North America, Europe, and China. Now we're starting to have, still not a representative sample, but at least less skewed. So there's, if you count the number of dots in that plot, there's 60,000 of them, and we threw in isolated sequences, and we had 4 billion of these small open reading frames, which for us means up to 100 amino acids. Oh, sorry, I did something I didn't want to do. Okay. So we keep the ones that are non-redundant, because if we only see them one time, we throw them away, we do a bit of filtering, and after this, we still end up with 1 billion of these. And we've put this out as a resource, and we have a preprint that Ichian has been working on. But now my research program for the next few years is going to be trying to understand, so what's really happening in this sort of underexplored part of the sequence space. And so as a first step, we went back and we looked for antimicrobial peptides. So antimicrobial peptides, as the name says, it's something that has an antimicrobial effect, either outright killing bacteria or often just inhibiting their growth. So we went back to these open reading frames, and we used a tool that we developed called Macrel to do this. And it's actually kind of funny because it's really like very standard 1990s machine learning. Sort of it's a random forest with hand-designed features, sort of very, almost retro computing. But the reason why we had to develop our own tool rather than is because so we don't get the highest accuracy. So here so our tool is there, Macrel. So it doesn't have the highest. Oops, sorry, didn't mean to do that. It doesn't have the highest accuracy, but it has the highest specificity. So it has the lowest number of false positives, because what we worried about is that we have all of this input. So we're filtering billions of sequences, and it's this sort of very classical thing. Like, so if you have a very small very small true positive rate, you're really risking it with false positives. There's the URL at the bottom with all the slides. So if you just want to take a picture of the URL, you get everything. There's no. All right. So we had a tool. We ran it through all of this, and what did we get? So as I said, we have this upper limit of 100 amino acids, but most of them are in this range of 20 to 50 amino acid range, which is also nice because it means we can synthesize them. So because we did all of this work, this took actually several years. There's computing, there's a tremendous amount of unglorious manual curation of data and metadata, all of this. And then the moment of truth is you go into the lab and, did you get it right or not? And so we were working with Cesar de la Fuente at UPenn, and we synthesized 100 of these, including 50 that were chosen completely at random. So it was completely unbiased there. And 79 were active, at least against one of the strains that we tested. So we were testing against the bad guys, so these escapee pathogens. And we also tested against the panel of commensals. And ideally, we would get something that kills all of the bad guys and none of the good guys. We don't really get that, but we do get that we have these sometimes very specific targeted effects where even strains of the same species have a different response. And that's actually a really interesting question that so we have this empirical data that shows that we don't really understand why, and we'll be trying to figure this out over the next few years as well. Why is this happening? So we also have in vivo data. This was done. So Marcelo Torres is the postdoc at UPenn with Cesar, and they were the ones leading this side of the effort. So then maybe we have a couple of stories. Well, but I'm just going to give you a little one, which is, so in real life, a lot of AMPs come from, you have a longer protein that's produced, and then part of it is cleaved, and now this peptide sort of has its own thing. Right? And whilst we were only looking at things that are encoded in the genome as their small open reading frames. But we were thinking, okay, but maybe there's some sort of cleavage at the genomic level, right? And so to test this, what we first of all, we aligned our short peptides against a catalog of full-length proteins. And what we saw is that, we got about 8% matches, but within those 8%, about a quarter exactly have exactly the same start codon. So they start where the big protein starts, and then somehow something happens, and there's a stop. And so here's, for example, just an illustrative example. So we have here there's this protein. It comes from Prevotella, and we see the alignments, and then these sort of orangey ones is these 3 metagenomic contigs, and there's been just a single point mutation that produces a new stop codon. So instead of a cleavage at the protein level, we have a sort of a genomic cleavage where this this is obviously not proof, but it's suggestive of this is one mechanism by which these things generate, and probably this is happening in evolution all the time. And if these peptides, if they're just garbage, they get filtered out, but maybe every once in a while evolution gets lucky, and now it generated a peptide that's itself functionally active in some ways. So we have a couple of other stories in the manuscript as well, but we also have everything's available. There's a website you can go to. So as I said, this was nice because in between getting the abstract in and today the paper was published there. So let me just say, to summarize, so that these small proteins, they're underexplored but vital. So there's billions of short sequences in the global microbiome. Some of them are really very highly conserved, and so actually Curtis was also mentioning this earlier. So we're in the process. So we do know that in these billions there's a lot of false positives there, but there are also these sequences that we see them repeatedly. We see them across phyla, across different habitats, across the world. No clue what they do, right? So really, there's no information there, and so this is now my research program for the next few years, and we've started looking at these antimicrobial peptides kind of as low-hanging fruit, easy to look at. They're also easy to assay because the assay, you can synthesize them chemically, and either they have an effect or they don't. And so with Marcelo and Cesar, we've been very successful at validating the approach, and we have even some preclinical studies. So Ichian, she's been doing the resource. Celio did a lot of the AMP work. This was done also with Per Bork in Heidelberg and Jaime in France. In Madrid, which is in Spain. I also want to thank So I was still in Shanghai. So the National Science Foundation of China funded the initial work. Now I'm in Australia, where the Australian Research Council is funding my work. So I want to thank them, and I want to thank you for your attention.
It was super interesting. I actually have a very simple question, which I didn't see you address, but I'm sure is in the paper. How do you prevent detection of partial sequences that you're getting from the metagenomes coming in? So you're talking about like truncation or potential truncations or stop codons, how did you prevent that?
So one thing we've done is, so if in the config, if you have an upstream stop that's in frame, that probably means that you're at least not in the middle of a gene. So we did a little bit of in silico QC. Empirically, what we see is that it does enrich. So this in silico stuff does enrich for having more good matches. But you also end up throwing away a lot of things. So that is still an open area of research for us. But yeah, that specific question you can look for in frame stops.
Really interesting. Thank you. I had kind of a two-part question. One is that, do you see like localization signals in these small peptides that are like retained? Like, how are they getting, like, if the thought is that they're antimicrobials, like how are they getting out? How are they affecting other stuff?
So how are they getting out was a question. So when I started working on this, I thought that other people would be able to answer for me. And I realized that it's actually, so there's a couple of mechanisms. So there's the standard, that they have a single thing, something else grabs them. But there's also, so some cases have been studied quite extensively where they're small and they kind of wiggle their way through the membrane, and they sort of diffuse. A lot of them, we frankly just don't know, and we don't know how to do this at scale. Is part of the problem. But yeah, it's. I also had that question, and I started asking the peptide people, how do these things get out? And I think the answer is nobody really knows.
Okay, cool. Yeah, you don't see any of them near like type 3 secretion systems or anything like that, or?
That's also a very good question. I have a student, and that's her PhD proposal.
Okay.
So maybe invite me for microbiome cozy in a few years.
Thank you.
Oh, sure. I'm actually curious, you started to allude to it, the evolutionary history of these short little, especially the ones that came from other larger proteins. Do you have any evidence of horizontal gene transfer? I'm just thinking in the sense of it's easy to have a little snippet go somewhere.
That's also a really good question. I don't have an answer. I do have another hat which I wear sometimes, which is horizontal gene transfer, but we don't know. I mean, so parts of it is, so we also don't have good methods for the. I mean, even trying to find homologs, it's just like the sequences are often just too small. For example, the other thing we can hypothesize and sort of wave our hands around is, it's possible that many of these are de novo sequences, because like you don't get the 800 amino acid thing that folds nicely by chance. You might get 20 amino acids that somehow randomly do something by chance, right? And so, but it's very difficult to investigate. But horizontal gene transfer, or whether they're present in plasmids, I mean, that was also a really good question. Also, and I'm not sure if it's you or someone else, but that was also a question from reviewer number 1.
Wasn't me.
Okay. Another question? Yes, please. Awesome talk. I'm curious whether you think that there's something about these 5' fragments that makes them more likely to be antimicrobial, or if that early stop codon behavior is true for most of the small ors.
Oh, that's a really good question.
Okay.
Yeah. I actually hadn't thought about it until you just asked.
I would check if it's true for all of them, because that seems like it would make sense, but I don't know either.
Yeah, that seems, yeah. I mean, so our model was that it was just that, so that you had the start was already there with the translation machine, where everything was already there, and then if you have a stop, now you're producing a small protein, and then this is what happens. But whether there's something more inherent to how proteins look at the start of, that's cool. Yeah, that's a very good question.
Okay,